regarding terminology of recombinant DNA technology. And there are different types of terminology which are involved in the process of recombinant DNA technology. Number one, foreign gene. The foreign gene, which is also known as the gene of interest, or gene have to be cloned, or transgene, or gene have to be incorporated. If you are define this process, then you can say the DNA segment with or gene which have to be transferred into the host cell and responsible for the desirable character. As you know that the different type of the genes encode the different types of the protein, and each and every character is governed by genes. So genes are responsible for the character, and DNA or genes are the storage material that they express the protein, and protein encode the character. Therefore, the DNA segment or gene which is going to be transferred into the host cell for desirable character, for desirable protein, is called as foreign gene or gene of interest, or gene have to be cloned. Second terminology is vector. Vector is too important because the vector is those molecule which act as a vehicle for the foreign gene. It is the molecule which carry the foreign gene into the host cell. So vector is a kind of the circular DNA, and it carry the foreign gene into the host cell, and definitely it is a circular DNA. So the source of the vector molecule is always the prokaryote, although some eukaryote like yeast also act as a vector molecule. Their DNA is also act as a vector molecule. So one of the important features of the vector is that the vector must be in circular in nature. Number two, it must have the autonomous replication property, and the autonomous replication property is due to the origin of replication. Now the foreign gene and vector is clear to all of you. So the construction of recombinant DNA takes place. The construction of recombinant DNA or chimeric DNA or hybrid DNA takes place by insertion of the foreign gene into the vector molecule. So recombinant DNA can be defined as a vector molecule containing the foreign gene or gene of interest into a recombinant DNA. So we can say it is a hybrid DNA molecule which consists of two unidentical set of DNA. So it is also called as chimeric DNA molecule, and whenever the recombinant DNA are constructed, this recombinant DNA is transferred into the host cell via the process of transformation. Number one thing is that whenever the foreign gene is incorporated into the vector, it must be that the vector molecule is digested by the restriction enzymes. The same restriction enzymes are used for the vector molecule as well as gene of interest for compatible binding.